Hi everyone, my name is Viola Chen and I'm a PhD student at Princeton University. Today I would like to share our work called Merlin for mitochondrial evolutionary lineage inference. Lineage tracing is commonly used for developmental biology. For example, in the case of embryonic development, we want to know the history of cell division during the development process. Specifically, we want to construct a cell lineage tree that tells us the lineage relationship across all the cells. In the past, people have used direct experimental observation for lineage tracing. In fact, the 2002 Nobel Prize for Physiology and Medicine was awarded to scientists who mapped out the cell lineage tree of C. elegans. However, this is only tractable for simple organisms as it was done with microscopy, and this is not tractable for more complex organisms such as human or even mice. In recent years, with the development of sequencing technologies, people have used somatic mutations for retrospective lineage tracing. However, there are a few challenges that come with this. First, the mutation rate of the nuclear genome is relatively low. It's only about 2.8 times 10 to the negative 7 base pair per generation. And this means that we have very few somatic mutations to work with. And secondly, these already very few somatic mutations are sporadically present in the genome, which means that if we want to use the entire genome, we have to do whole genome sequencing, which can be very costly. Alternatively, we can do targeted sequencing of the genome, which will give us only a very small subset of the somatic mutations to work with. One commonly overlooked fact is that at least 5% reads in single-cell RNA sequencing experiments are of the mitochondrial origin. In fact, mitochondrial reads are being picked up in almost all the single-cell sequencing experiments from single-cell DNA to RNA and the tag-seq. And mitochondrial mutations have certain properties that overcome the challenges that come with lineage tracing with somatic mutations. So first, the mutation rate in mitochondria is about 10 times higher than that of in the nuclear genome. So this means that we have a lot more mutations to work with if we look at mitochondrial mutations. And secondly, the mitochondrial genome is much shorter compared to the nuclear genome. It's only about 16,000 base pair long. So it's much cheaper if we want to do mitochondrial enriched sequencing. Therefore, we propose to use mitochondrial mutations as natural markers for lineage tracing. However, mitochondrial mutations has its own unique set of features that we must pay attention to. Specifically, mitochondrial genome has this phenomenon called heteroplasmy. Unlike nuclear genome, where there are only 2 copies being a pair of chromosomes, each cell has hundreds to thousands of mitochondria in them, and those mitochondrial genome in every cell is not always homogeneous. Oftentimes, there are one or multiple mitochondrial clones with distinct mutational signature present in every cell. And for example, here we can see that the blue clone and the red clone could be in like a 70-30 proportion. So from single-cell sequencing experiment, we're able to obtain the variant allele frequencies of the mitochondrial mutations just by taking the proportion of alternate reads over total reads. And we want to ask the following question. Can we infer a mitochondrial clone tree, which tells us the mutational history of mitochondria, like the tree shown on the left, and the cell lineage tree, like the one shown on the right, just from this single-cell data, specifically looking at the mitochondrial mutations. And our idea is that from the frequency matrix F, with rows being cells and columns being mutations, we want to factorize it into a mixture matrix U, which tells us what mitochondrial clones are present in every cell, and a clone matrix B, which tells us what mutations are present in each clone. It has been shown that there's a one-to-one correspondence between the clone matrix B and mitochondrial clone tree shown on the right of the screen. And here we make a simplifying assumption that this mitochondrial clone tree is a perfect phylogeny where mutation is gained once and subsequently never lost. This problem is actually not new. For this factorization problem, there have been multiple methods developed in the past 10 years that are able to factorize F into matrix U and B. So what is new here? In fact, there are multiple ways to factorize F equals to UB, and that is the problem. So in our case, the rows in the mixture matrix U corresponds to single cells. And we know that these single cells can be put into a cell lineage tree. Okay, there are lineage relationships across the different cells, which also lies on phylogeny. So we proposed a new problem called the nested perfect phylogeny mixture problem, NPPM. So given a frequency matrix F, we want to find factorization F equals to UB if one exists, such that the mitochondrial clone tree from B and cell lineage tree from U are both perfect phylogenies, and they have to be concordant. What does it mean to be concordant? So as mentioned earlier, we have a cell lineage tree T, where we assume the daughter cells inherit all the mitochondrial clones from the parent. And we also have the mitochondrial clone tree S, which is a perfect phylogeny. And since mitochondria are present in the cells and inherit through the process of cell division, if one mitochondrial mutation happen on top of another or after another, we would expect the cells that contain the latter mutation to be a descendant of a cell that contains the former mutation. And this has to be the case for each pair of such ancestral descendant relationship. And this is what we mean by concordance. We can also view this as a nested phylogeny, where the phylogeny of mitochondrial clones is nested within the phylogeny of cells. In our paper, we formalized this as a theorem which states that a cell lineage tree T and a mitochondrial mutation tree S are concordant if and only if the partial order, which is the ancestral descendant relationship induced by the cell lineage tree T, is an extension of the partial order induced by the mitochondrial clone tree S. So to give a clearer picture, here's an example of one way of factorizing F equals to U and B. And I claim that these two trees are not evolutionary concordant. So why not? So here we have the mutation by a red square arise on top of the clone with mutation with a triangle. And we would expect that the cell containing the square mutation to be a descendant of a cell that contains the triangle mutation. However, this is not the case here. We say that for a mitochondrial clone to arise in the cell must contain its ancestor, which is not the case here. And if we list down the partial order induced by the two trees, we can see that they're not concordant. For the same frequency matrix F, here is a different way of factorizing it into U and B. And here, the two trees are evolutionary concordant, as we can see from the partial order listed here. So now we have formalized this problem, nested perfect phylogeny mixture, and we want to know that which frequency matrices admit a solution to this problem, or do all the frequency matrices do? In our paper, we characterized conditions for a frequency matrix F to admit a solution to the NPPM problem. And these two conditions are necessary and sufficient, and we also show in our paper that checking this condition is NP hard. In reality, most sequencing data sets come with noise. So instead of posing this as a factorization problem, we frame this instead as a optimization problem where we try to minimize the L1 distance between F and UB. And we solve this with a mixed integer linear programming approach implemented in our package Merlin for mitochondrial evolutionary lineage inference. To benchmark the performance of Merlin, as there's no other methods that directly uses the mitochondrial mutations for both like cell lineage tree inference and mitochondrial clone tree inference, we benchmarked Merlin with method that does either the cell lineage tree inference or the clone tree inference. On simulated data, we first look at the error in the inferred cell lineage tree. So this is the average Hamming distance in binarized U matrix. And we have shown that Merlin outperform all the other methods on this metric. We also look at the error in the inferred mitochondrial clone tree, where we compare the normalized parent-child distance in the inferred mitochondrial clone tree and shown that Merlin performed the other methods that are able to give a clone tree output. Next, we use Merlin to look at a public in vitro gastric cancer dataset that was published in 2020. So this dataset has about 5,000 cells, and we're able to identify 14 informative mitochondrial mutations. And here is the mutation tree inferred by Merlin. And we noticed that one of the branches in our tree corresponds to cells that are enriched for a deletion in the Q arm of chromosome 4, giving us some confidence that we are identifying a tree that is biologically plausible. We also compared the other methods on the same data set with Merlin just to see the difference. So on the right, we have the mitochondrial clone tree inferred by a pear tree. It is very similar to the one inferred by Merlin. However, unlike Merlin, which makes sure that the cell tree and clone tree are concordant, we found that almost half the cells in the mixture matrix from pear tree are evolutionary discordant. We also tried other methods such as CITE on the same dataset, and here is the mutation tree identified. The issue here is that on two separate edges, highlighted in red, which stands for two independent mutations, these two mutations show a very high Pearson correlation in the variant allele frequencies. So what this means is that these two mutations are likely to be part of the same mitochondrial clone that are inherited together through cell division and mitochondrial replication. And that's the most plausible explanation for this high Pearson correlation between the variant allele frequencies. And therefore, it's more plausible to group these two mutations into a single edge, like Merlin did. We see something very similar for another method, physics, where we also observe this high Pearson correlation on two separate edges that have a high Pearson correlation and should belong to the same mitochondrial clone. And therefore, we think that as Merlin takes into consideration the complexity in mitochondrial mutations and its inheritance, we're able to produce more biologically plausible results on the real data. In conclusion, we introduced the nested perfect phylogeny model for the evolution of mitochondrial clones in the developmental system. And we characterized the frequency matrices that admit nested perfect phylogenies. Lastly, we implemented our method in Merlin, which jointly infers the cell lineage tree and the mitochondrial clone tree from single-cell sequencing data. Lastly, I would like to thank everyone in my group, and especially my co-author, Dr. Palash Seshito, who will be joining Virginia Tech as an assistant professor this fall. And he's a great mentor, and he will be looking for students. If you or someone you know is interested, feel free to contact him. And we have the QR code for our paper and GitHub. Thank you very much.
This on now? Yeah, got it. Okay, so my question has to do, I like your model. but with more real-life data. So when you do single-cell analyses, you have this one snapshot in time, but mitochondrial dynamics are really dynamic. Do you know how well your lineage tree method performs under different types of cell stress that might cause more, say, fission, fusion, proliferation, any of those perturbations?
Thank you for that question. I
Kind of more
actually,
just philosophical.
yeah, we actually, I personally, like, I read a lot of papers on mitochondrial fusion and fission. The issue is that there's very limited data or like ways we can validate just whether fusion or fission take place. And personally, I would like to see more data sets on mitochondria just under different stress, and to be able to look at how they affect the changes in mutation. But yeah, we weren't able to look at it.
Maybe in the data set that you analyzed, were you able to look at also by genotype from the nuclear genome, like maybe what type of cell they might be or what state they might be in from non-mitochondrial signals?
So we're not able to identify enough clones from the nuclear mutations or copy number.
Okay.
Yeah.
Thank you.
Oh, nice talk. So I'm curious about the computation numerical limitations. How many cells can you manage with this method?
Thanks for the question. So the largest number of cells we run is for this data set with about 5,000 cells. Yeah, and they finish in like a few hours.
Hi, thanks for the talk. I was just wondering, you mentioned, that the mitochondrial genome, which is much smaller, has much higher mutation rate. I worry about like infinite sites assumption violation. And do you have any idea, is this a big problem or not a problem for your setting?
Thanks for the question. It is something that I personally considered. This is the first method that like tried to use mitochondrial mutations for this tree, and I think there are like finite set models there, and personally I would be interested in exploring that.
Okay, did you do anything to like estimate how often this might happen, or is that sort of unknown at this point?
I did some numerical calculations about like how often this happened, but this like the current model assume infinite site assumption.
Okay, thank you.
Yeah.
Does this method work for standard 10x single-cell RNA-seq, or do you need to specifically enrich for mitochondrial reads?
Thanks for the question. This method, so basically from the RNA-seq sequencing, we first like call variants, and then we use the method called mQuad to identify mitochondrial mutations that are informative. So this works for any data. It's just whether we're able to identify enough informative mitochondrial mutations. On data sets that are enriched for mitochondria, it performs slightly better. We have a higher coverage, and we're able to find more informative mitochondrial mutations.
Yeah, so fascinating talk. This is sort of, I guess, a follow-up to some degree of the question about mitochondrial dynamics. But even in the absence of, sort of stress conditions or something, it strikes me that you have a large mitochondrial copy number in some cells. Your initial mutation occurs in a single copy, and then you have a heteroplasmic state. And I guess I'd just like to hear you speculate about whether that's a problem. Have you considered that?
Thanks for that question. So if we look at the tree we shown here, so for our model, for the root node in the mitochondrial clone tree, we assume it's a dummy root node, and you see this hexagon here that stands for a group of mutations. So basically in our model, we allow this kind of like a dummy root node with an edge with a group of mutations that kind of leads to an initial kind of first initial state, if that makes sense.
Yeah, I'm still trying to, I think, figure this out. So the one place where you're having multiple mutations is along that root node. So what you're saying is you could interpret that as a heteroplasmic ancestor. Am I
Yes.
understanding?
Yes.
Okay. Can you interpret anything else as heteroplasmic in this?
So this is a mutation tree, and we assume heteroplasmy
Okay.
when, yeah, just in the cells.
Okay, that makes total sense. That's a great, thank you.
Thank you.
Hi, thank you for your presentation. Does it have to be mitochondria? Can I just get really good ISMB list and run this algorithm?
It doesn't have to be mitochondria. So with nuclear, like with confident like nuclear mutations, there are other methods, existing methods that already uses the nuclear SMVs for lineage tracing. So I think the highlight of our method is the nature that we make use of like heteroplasmy, and therefore mitochondria.
So then without the heteroplasmy, so it'll be just diploid and haploid and it is complete. Would it, the benchmark result, if you have tested, was it comparable to the other methods or was it still better?
We did not compare our method with others on nuclear mutations. Thank you.